Hi everyone, my name is Toluleke Famuyiwa. I'm a PhD candidate in integrated biology. The title of my presentation today is Drug Release Studies of SC514 Polylacticogolic Acid Nanoparticles. This is my presentation outline. Background of study. Prostate cancer is still a major concern because there is still high incidence of death from prostate cancer recorded in America. Because of this, drug resistance has been implicated as one of the major causes of this death from prostate cancer patients. One of the ways that we can reduce drug resistance is to use nanoparticle drug delivery system, in which case we are placing the drug, in this case SC514, inside nanoparticles, such that these nanoparticle can specifically target the prostate cancer cells with minimal effect on the healthy cells of the body. In addition to that, we can also coat the surface of these nanoparticles with ligands that are specific to the surface or the cell surface markers on the prostate cancer cells, as explained in this diagram. The objective of this study is to place SC514 inside PLG nanoparticles, conjugate the surface of the PLG nanoparticles with fat and nuclear factor kappa B, such that we can engage in controlled drug release and increase the therapeutic efficacy of SC514. Because one of the reasons why most of the drug does not really work very well is because there is no control release that can help to reduce drug resistance. This is the aim of the study. The aims of the study follows the same explanation as before. This is the hypothesis of the study, that if we improve the therapeutic efficacy of SC514, then drug resistance can be reduced in prostate cancer cell. In order to test this hypothesis, we went through all these steps. We performed MTT assay. We used immunofluorescence assay to measure multi-drug resistance in the prostate cancer cells. We designed a nanoparticle using single emission method with conjugation of the surface of nanoparticles. We also engage in liquid chromatography mass spectrometry instrumentation so that we can quantify the SC514 inside the nanoparticles. We explored new methods to be able to quantify the SC514 inside nanoparticles because LC-MS is a very expensive method and it's not readily accessible. The results from our study indicated that the SC514 PLG nanoparticles without conjugation exhibited burst release, as indicated in the orange colored graph. Then when we conjugated the surface of these nanoparticles with nuclear factor kappa B, in fact, we saw that the burst release was removed, as indicated in the purple colored graph and the blue colored graph. Also, we explored new methods to be able to quantify the SC514 inside the PLG nanoparticles. We observed that these three methods, colony assay, transfer assay, and wound healing assay, were very effective to quantify the SC514, but these three methods were not as effective or as sensitive as the standard method. And we observed that the wound healing assay was the most sensitive to quantify the SC514 that was released from SC514 PLGA nanoparticles. This is the picture taken from the colony forming assay, looking at what happened from day 0 in figure A up to day 7 in figure I. We quantified the number of cells within the colony and we used that to generate our drug release curve. Similarly, we perform transfer assay as an alternative method to measure the SF514 inside the nanoparticles. The cells that migrated to the bottom of the transfer were quantified, and we used that to generate the drug release curve. Also, we performed wound healing assay as an alternative method to quantify SF514, and as the width of the wound is closing, we calculate that change in width. We use that to generate drug release curve as we move from day 0 all the way to day 6 in this diagram. All right, we also performed confocal microscopy, and with this we were able to see that the PC3 cells were taking up, absorbing the nanoparticles as time passes. It's a time-dependent absorption with respect to these nanoparticles we're talking about. Also, we perform inverted microscopy to be able to know the impact of the drug release on PC3 cells, and we saw that when we did not conjugate the surface of nanoparticles, there was burst release. Most of the cells were dying even at day 1, at day 2 in figure E and F. In figure A, B, C, and D, when we conjugated the surface of the nanoparticles, we saw that there was hardly any impact on the cells on day 1 and day 2. PLGA polymer was used as a control. We also have treatment from free SF514 in this figure. Now, we perform MTT assay to know the impact of the drug release on the cord blood cells, and we saw that very minimal effect of the drug release. In this case, SF514 was observed in most of the treatment, polymer used as the control. We performed a similar MTT assay analysis on the PC3 prostate cancer cell. The result from this MTT assay indicated that PC3 cells were more impacted by the treatment, especially in figure D with the SF514 PLGA formulation compared to what we have in cord blood cells. In PC3, we have the Warburg effect, which makes PC3 more predisposed to treatment of this type of drug compared to what you have in cord blood cells, which is a typical normal cells of the body. We also performed immunofluorescence assay to quantify the number of drug-resistant cells in treatment that we have. We observed that the combination treatment inside nanoparticles impacted the lowest number of multi-drug resistant cells compared with a situation of just the 2 drugs without nanoparticle. Similar results in PC3 was observed in these D145 cells compared to what we have in the last slide. Discussion. Here we are able to see that putting the drug SF514 inside the nanoparticle was very effective to reduce drug resistance, also to reduce the number of cells that are living. And we can see that this can be extended to other studies. In conclusion, development of this drug delivery system can be used for other types of drug, in which case we can now use less amount of drug and less amount in the frequency of drug application so that there's less side effect to the healthy cells of the body. This can be used for designing other types of drug in this study, as can be used for other studies. These are my references. My acknowledgement goes to my committee members, my lab members, my contributors, and also the various funding sources that was used for this study. Thank you for listening. Bye for now.